So in this part, I will show how to process the data obtained from lipid nanoparticles, where I calculated the incorporation efficiency of the RNA. So for that, I first import the data. Here I have files with different ratio between RNA molecules and lipid molecules. So 6 means that I have the most RNA per lipid. 24 means the opposite one. And here also there is a blue channel 1, which is from the lipid dye. And the orange one is the red channel, which comes from the RNA labeled by Alexa 647. So you go to peak analysis. And in peak analysis, for example, in first of all, in NP6, you have to, well, you can put together all of them. And then what you do, you also select here, detect by channel 1, because right now we want to detect by lipid dye and to show whether every particle that has lipid has RNA. So here you see this histogram, and histogram has clearly two populations. One population is over here, and the second population is over here. So this histogram is calculated by the same GP formula, which means that when you have population over here, it has a lot of signal from the green channel and very little signal from the red channel. So this means that this is the population that corresponds pretty much to the empty particles. So a lot of signal from the green channel means lipid dye, and little signal from red channel means no RNA in there. And the second population is over here, which means that, yeah, it has both lipid and RNA. And then when you move, for example, from 6 to 24, let's move to 9 first. So when you move to 9, again, I will move here to 40 to have all of the repetitions together. Here, channel 1, you see that this second population is smaller. And then in 12, you have smaller, and in 24, you have the smallest over here. So right now it's going to be visible. Yeah. And now, what we also want to calculate, we want to calculate how much particles actually have RNA. So here in total, we calculated 15,323 events, which corresponds to 15,323 particles. And now, instead of detecting particles by channel 1, I will detect them by both end channels. Both end means that in both channels, particles are above the threshold. And here it's prominent because you see that instead of having two populations, immediately you have only one population, which is around 0. So we take into account only particles that have both lipid dye and RNA, so particles that are full of RNA. And the total count is 6,844. So now what we need to do, you just need to divide this by 15,323, which is something like 40%. But let me make sure. Yeah, it's 44%. So 44%, 45% incorporation rate, that's what we actually have for nanoparticles, lipid nanoparticles at 6, and in 24, there is very little also RNA per particle, but you see that when you have both, it's actually just this population which is closer to 1 because there is less RNA per particle, but we have here 4,723, and then when I go to here it's 14,000, so it's something like 30-35%. Yeah, that's how we do it. That's how we calculate how much particles are full of RNA and how much RNA you can have per particle. You also can calculate, but for that you need to do the proper studies of molecular brightness of RNA molecules. So in this case, we had for every RNA we had one Alexa 647, and you need to calculate the molecular brightness, but it wasn't that easy in this particular case because RNA aggregated in solution, and we couldn't actually know the brightness of one RNA molecule. But if you succeed to do such a calibration, you can also convert these histograms into the not only GP but kinda number of RNA or some other value that will represent them.